We're actually on the UCSD campus, the Institute of the Americas, for the second annual UCSD Center for Chronobiology Symposium, and it's actually called From Cells to Clinic. One of the speakers, we've just done a panel, but I want to find out a little bit more about how you got into all of this business, Andrew Millar. And you're technically at the Institute of Molecular Plant Sciences, School of Biological Sciences, University of Edinburgh. But really, what this is about is the Centre for Systems Biology at Edinburgh, right?
Yes, that's right.
And biological clocks. I just looked at the brief bio here. And it starts off with the European School of Luxembourg. So you actually didn't start in the English school system as
No.
No?
That's right.
How did that all start?
So my father worked for the European Parliament and the European Parliament, as you might know, viewers might know, it meets both in Strasbourg and in Brussels, but all of its staff live in Luxembourg. So there's a mass migration across the train network when the MEPs go to one or the other. But several of the locations which have a lot of European staff have these so-called European schools, which are a marvelous educational Tower of Babel. So the French in the French section are taught in French, and the Germans are taught in German, and the Italians in Italian, and so on and so forth. And so there's an English section which started in 1974 when the UK joined the European Union. So that's where I did my schooling in primary school and secondary school, and one comes out being good at languages.
So, but was there a plan here? When did you first know that you wanted to do science? Because your father's obviously a diplomat. I suppose that is an art and a science. But your mother a scientist? Or
no, there are medics. There are medics in our family, but no scientists.
Yeah.
So it was. I fractured my skull when I was 10, and the family went on a Caribbean holiday to visit one of our relatives. Because I was not terribly well, I was left alone in a house in Tobago with not very much to do, but a long bookshelf full of the back numbers of National Geographic. So actually, as I ploughed through National Geographic issue after issue, I became fascinated with biology, with marine biology specifically. And I imagined that I would learn to scuba dive and get a suntan. So neither of those things worked out.
Right. But take the trajectory a little bit further, because it's always very interesting for young scientists, particularly, to figure out how other people got into this business and why and so on.
So thereafter, I was pretty sure by secondary school that I was going to go into biology. And at the time, biotechnology was rising in the early 80s, this would be. So I went to Cambridge as an undergraduate, and my interest in plant science was definitely sparked by a particular academic in Cambridge at that time, a guy called Tom Abrice, who worked on carbon metabolism in plants, and particularly on how plants determined how much carbon to store as starch and the enzymatic mechanisms that allow them to partition carbon. So that just generally sparked an interest in plant biology, but I trained as a geneticist in my undergraduate days. So I wanted to go to a molecular biology lab, which by the late 80s, plant molecular biology, making transgenic organisms, was the best way of understanding plant regulation. So the story goes that I went to a lab in New York City, a place called the Rockefeller University, to a laboratory of Professor Namhai Chua, who is a very distinguished Singaporean scientist who'd worked in Rockefeller for many years. He was then the only plant lab in what is otherwise a biomedical institution. I went to Nam's lab to work on root nodulation. So root nodules are the structures in legume roots which fix nitrogen. So peas, beans, alfalfa fix nitrogen in root nodules. And it was a marvelous genetic system because in the nodule, the bacterial genetics interacts with the host plant genetics. And so it's for geneticists, it's very attractive on a fundamental level, but it also has a very important applied aspect. Unfortunately, that didn't work out at all. In fact, it didn't work out pretty much from the first week that I was in the lab. And so, quite by chance, Professor Steve Kay was there as a postdoc.
Who now runs the
who's now the Dean of Biology here in UCSD. And Professor Chua suggested that I should go and talk to Steve, and maybe I could come up with a project with him. Now, Steve had just stumbled across circadian regulation, 24-hour regulation of gene expression, in a project where he was basically working on light signaling and how genes respond to light. So he and another Hungarian scientist working in the lab had done what were meant to be replicate experiments. Steve's experiment and Ferenc Naj's experiment should have given the same result. But Rockefeller had a very good social scene, and going downtown was quite a common occurrence. So as it happened on that particular occasion, one of them got their experiment done before they went out, and then one of them had to wait and did the experiment after they came back from a night out. The two experiments were quite different in their results. After some mutual recrimination, the story goes, they determined that only the time of day differed between the two results. The results were different because the experiment had been run at different times, and that sparked for both of them a long-standing interest in the circadian clock. And so I was the first student to arrive after that discovery, and so I picked up the circadian clock project. I was very lucky, because at the same time some groups elsewhere were developing a new species for plant molecular genetics. So this is a little weed. It's called Arabidopsis thaliana, is the Latin term. It doesn't really have a good common name, but mouse ear cress is sometimes referred to as mouse ear cress. So in 1988, this was just becoming a feasible model. It's now the main genetics. So I started on clocks in plants, and specifically in Arabidopsis, almost from the start of both the clock field in the molecular era and also at the start of Arabidopsis genetics. And so it was a very lucky coincidence that those two initiations started off and I was in the right place at the right time. And so I've been working on circadian clocks in Arabidopsis ever since.
Interesting how many times when I ask these questions of people, there's often a serendipitous story comes in. I ask people, there was the book, but you would recognize, Advice to a Young Scientist by Peter Medawar. And then there's also Advice to a Young Investigator, of course. So having gone through that, having realized how serendipitous all this sort of stuff is, and what would your advice to a young scientist be? It sounds like part of it would be choose your mentor wisely as well.
Yes. When I went to ask my mentors in Cambridge which labs I should look at for a PhD position, I was very taken aback. I went into the room and one of them turned around and said, well, if you want to be famous, then you should go and work with Namhai Chua. And it hadn't really ever occurred to me that I might want to be famous, and that wasn't why I thought about doing science. But of course, science is a social process, and successful scientists are successful because they manage to convince a large number of other people that their ideas or their results have importance. And so a certain amount of notoriety, at least, is part of the scientific process. And that was something which I was being taught at an early stage. But that was advice that I maybe wasn't quite ready for at that time. It turned out to be very good advice.
What would you pass on yourself now, though, to people coming into a lab?
I would say that choosing your mentor is important. Giving yourself enough time is important. I was very impatient when I was starting my PhD, and in fact I benefited enormously from spending 6 years doing a PhD. Use the freedom that you have. It doesn't feel as though you have a lot of freedom as a PhD student, but you do. And as science, the process of doing science is now very pressured from the moment that you start your first postdoc, really. A PhD is a very, very valuable time.
The whole field of plant biology, we mentioned in the panel discussion that we did, I often hear people saying how pathetically small the amount of that field is, yes, whereas it actually seems to have an enormous importance and growing importance. I mean, am I wrong here? Or
No, I think you're absolutely right, but you would expect me to say that. But the reason I say it is that plant scientists are being asked to solve two of the very biggest societal challenges, food security and bioenergy. Now, it's not just plants in bioenergy, but plants are still a large proportion of the proposed solution to a bioenergy strategy. And the amount of funding that we're being asked to do to tackle those enormous challenges on is nothing like enough. I think the Royal Society report 2 years ago suggested that it would take $100 billion over 10 years, which it sounds like a very large amount of money, but compared to medical research spending, it's you have to get plant science up to something like that scale if you're going to have a chance of dealing with producing crops which are tolerant to new stresses and new combinations of stress, if you're going to understand plant metabolism enough to engineer new fuel sources or new biomass sources. And also, there's an enormous skills agenda in plant science. So we've lost a lot of academic activity which used to happen in areas related to soil science and agronomy. Now, soil science and agronomy are areas which one can apply much more rapidly than the genetically based approaches of crop breeding or GM. And speed is now of the essence, because the crunch point for food security may only be 20 years away. And it takes 15 years to breed a new line of wheat. So we don't have much time. You mentioned GM and genetically modified organisms, huge furore certainly in UK, yes. Bit less so here perhaps, I don't know. But how do you now feel that the climate is in terms of those sorts of things? This is very much a sense of how science is communicated itself by scientists themselves also. Yes. Absolutely. Well, there are many factors that go into that. Scientists have the opportunity to communicate within a context which is framed by a number of other stakeholders. So you will no doubt know that I work in GM-free Scotland, right, thanks to the Scottish Nationalist Party, who are currently in the Scottish Executive. So, as you might expect, I have enormous difficulty with a dogmatic position taken at a time when we face unprecedented challenges, and particularly a real risk that unexpected events will happen, and in the climate and pest dimensions. So we know that unexpected things will happen, and by removing the possibility of even testing GM crops on a large scale, it's clear that one of the potentially powerful tools in our arsenal is being removed from us, and this seems... Well, speak to this thing, this science and society issue. One of the questions I often ask people, many of these answers up on the Science Network. President Obama, when he came in, said his administration would restore science to its rightful place. No coordinates were given. What do you think is the rightful place of science? What is the rightful place of science? I think the Enlightenment never was a popular movement. Right? It was a movement that was led. And the Enlightenment project has some way to go. So, however, I think we're living at a time when science is allowing a new sort of consciousness, where many people become aware of what the population, the human population around the globe, is doing as a whole. Now, that's never been possible before. So this is a sort of thinking where many more people have to take into account the actions of humanity as a whole. That's only possible through science. And all of the ideas which come about human impacts come through the various different disciplines of science. So in those sorts of ways, the new drivers from environmentalism, issues around globalization, which are not environmental, they're much more economic and political, these are factors which have not previously entered political debate and social consciousness. So I'm not sure that we, as scientists,